Friends, welcome to Subhajit Biomedical YouTube channel. Today we will discuss about inborn errors of metabolism. So what is it? Let's begin. Inborn errors of metabolism from a large class of genetic disease. It means it is a genetic disease where gene is involving congenital disorder of metabolism. What is your metabolism? Metabolism means the uses or utilization of glucose, carbohydrate, protein, fats to metabolite products. The majority are due to defects of single genes that codes the enzyme that facilitates the conversion of various substances or substrate into other products. Inborn errors of metabolism now often referred to as congenital metabolic disease or inherited metabolic disease. That means one gene or one enzyme may be deficit from your birth, from when you are staying in your mother's womb. So the term coined by British physician Archibald Garrett, and he is known what for the prefigure the one gene and one enzyme hypothesis. And on his studies on the nature inheritance alkaptonuria, and his seminal text Inborn Errors of Metabolism was published in 1923. The inborn errors of metabolism are a heterogeneous group of disorders that may be inherited or a result of spontaneous mutation. This disease involved failure of metabolic pathway involved either in the breakdown of storage of carbohydrates, fatty acid and proteins. So what is the epidemiology? Inborn errors of metabolism occurs 1 in 2500 births. That means one baby or one person are affected throughout 2500 people. And next, inborn errors of metabolism. The diagram, you see the diagram. In case of inborn errors of metabolism, if you have gene mutation, then gene can produce protein by mRNA. That protein would be abnormal or non-functional protein, and this non-functional protein produce non-functional enzyme. The enzyme is very non-functional or it is damaged. By that non-functional enzyme, the metabolites products would be abnormal. This is the normal diagram and normal pathophysiology of the inborn errors of metabolism. What is the molecular basis of inborn errors of metabolism? Inborn errors of metabolism, that means the metabolic pathway. You think suppose metabolic pathway is a series of biomolecule reaction where each reaction is controlled by a specific enzyme, and according to one gene and one polypeptide hypothesis, a particular gene control particular polypeptide chain. Therefore, if there mutation in the particular gene, there are also reflection in the chain of the polypeptide. And you already know enzyme may be consist of single polypeptide because enzyme consists of protein and enzyme is protein in nature. So their enzyme may be consist of single polypeptide or more than one polypeptide. Therefore, if there is defect in the particular gene by mutagens, then the concerned enzyme will be defective, and thereby a specific metabolic reaction in that metabolic pathway is interrupted, which leads to inborn errors of metabolism. And next, what are the agents for the development of pathophysiological symptoms to IAM? What is the agent? Number one is due to defect of the gene by mutation, the concerned enzyme would be affected, and thereby metabolic pathway would be interrupted of that step. As a result, the substrate of the previous step accumulated in the cell. Such excess accumulation of non-functional substrate may lead to pathological symptoms. 2. Due to interaction of the particular gene, the metabolic pathway, the product of the sites or the end product of the sites would be decreased. and due to deficiency of such product in the cell, pathological symptoms also develop. Number 3, due to accumulation of substrate, alternative metabolic pathway opening and become functional, that generates some biomolecule which are not tolerated by the cell, and this result develop the pathological symptoms. This is the molecular pathway. And number 4 is due to interruption of specific metabolic reaction. In that metabolic pathway, accumulation of substrate may induce feedback inhibition of the enzyme which are appear in the at the prior steps. As a result, more than one types of substrate are accumulated, and several new metabolic pathways are opened that generate non-traditional biomolecules that lead to pathological symptoms development. So what are the types of IEM? Disorder of the carbohydrate metabolism. IEM should be involved in carbohydrate metabolism. The example are glycogen storage disease, glucose 6-phosphate deficiency disease. It would be amino acid metabolism, disorder of amino acid metabolism. That means phenylketonuria or glutaric acidemia type 1. Maybe disorder of organic acid metabolism, alkaptonuria. Maybe fatty acid disorder, medium chain acetyl coenzyme and dehydrogenase deficiency. And disorder of porphyrin metabolism, acute intermittent porphyria. And classification may be amino acid phenylketonuria and homocystinuria and tyrosinemia, lipidosis, Tay-Sachs disease, Gaucher disease, and metanometric leukodystrophy.